Dear students, now I will represent to you lecture Skeletal Tissues. First of all, I want to say about types of cartilages or classification of cartilages. In general, recognize three types of cartilages: hyaline cartilage, elastic cartilage, and fibrocartilage. Its cartilages differ by types of composition, extracellular substance, types of fibers, and in result, specific function. Similar to all types of connective tissues, cartilage composed of cells and extracellular substance or extracellular matrix. And in extracellular matrix present specific elements. First of all, it's fibers and ground substance. Cells in cartilage represent by two types of cells, chondroblasts and chondrocytes. Which is structure of cartilage in general? In general, cartilage covered by perichondrium. It's a superficial layer of cartilage which include two sublayers. More deeper layer has name vascular part or vascular layer, and more outer layer has name cellular layer. In cellular layer present chondroblasts, and more deeper part include more blood vessels. In the central part of cartilage present chondrocytes with extracellular substance. In extracellular substance present fibers, specific fibers to every type of cartilage, and ground substance. Chondrocytes include more rough endoplasmic reticulum Golgi apparate because its cell produced more protein. Protein formed extracellular matrix or extracellular substance. Very interesting question about vascular supply or blood supply of cartilage. Blood vessels present in cartilage just in perichondrium, in central part of cartilage not present blood vessels, and all nutrition material and all elements from blood can diffuse from perichondrium to more deeper part of cartilage. And in central part of cartilage present chondrocytes, and chondrocytes takes specific nutrition material and form active process production of proteins. Hyaline cartilage is the first type of cartilage which I will represent to you on this lecture. Hyaline cartilage present in embryonic period, in adult present. It's a common type of cartilages. Cartilage has white or light blue color and jelly-like structure. Inside present chondrocytes. Chondrocytes surround of collagen fibers, and groups of cells of chondrocytes form special isogenial groups from 2 to 8 cells, and every type of group of cells surrounded by fibers and form a vacuum. Ground substance in cartilages include GAG, mostly chondroitin sulfates and hyaluronic acid, and small amounts of keratan sulfate and heparan sulfate. Also proteoglycans and proteoglycan aggregates, glycoproteins too, and some elements of next large molecules, protein, fibronectin, chondroitin, nectin, and fluid. And fluid, it's include water more, near 70%. It's good picture show to you position of large molecules in cartilage, and chondroitin sulfate form chains, proteoglycan and glycoproteins, and form special cross-link matrix, and it's very important to form jelly-like consistency of cartilage. Collagen fibers type 2 present in hyaline cartilage. On its slide, good picture show to you position of isogenial groups. In lectin may be present from 2 to 8, sometimes 15 cells in one group of cells. Also present perichondrium present in peripheral part of its cartilage on picture, and you can see light color of cytoplasm in cells, and in perichondrium present more collagen fibers and cells, chondroblasts. Chondroblasts lie in perichondrium in more deeper part visible in this picture, and chondrocytes include in central part of cartilage matrix and surround of collagen fibers and ground substance. Very important question about histogenesis. All cartilages derived from embryonic mesenchyme, and in early period of formation, mesenchyme cells lose prosthesis, stay more oval, and form prechondroblasts. Next, chondroblasts form produce collagen fibers and ground substance, and in future form chondrocytes. Its good scheme show process formation of cartilages. On first A part of its picture present mesenchyme cells, which joined one by prosthesis and form more cells in mitotic division period. Next, type part B. On part B present mesenchyme cells which form oval shape and lose prosthesis and stay more form groups of cells. The next C. It's a precursor of chondrocytes, prechondroblast, and then chondroblast. Chondroblast cells to divide and form isogenial group. Why we have name isogenial group? Because one cell, chondroblast, in future form isogenial group. It's a group from 2 to 8 or sometimes 15 cells in one group. And isogenial because all cells formed from precursor one cell. In other words, one cell form group of cells. Elastic cartilage has yellow color and very flexible because include more elastic fibers. Elastic fibers surround of groups of cells. Isogenial groups of cells form parallel columns, and inside in one group can be 1 or 2 or 3 cells. It's photo from a histological specimen after staining of orthine. Elastic fibers surround of cells and form isogenial group. After staining of orthines, elastic fibers have brown color or dark red color. Fibrocartilage is special cartilage which lie between vertebral discs and characterized by elements from hyaline cartilage and from elastic cartilage. In fibrocartilage, not form isogenial groups of cells, and cells lie between collagen fibers. Collagen fiber type 1 predominant in fibrocartilage, opposite of hyaline cartilage. In hyaline cartilage, present collagen fiber type 2. Students, it's photo from histological specimen after staining of hematoxylin and eosin. Collagen fibers form parallel layers, lie between cells. Cells lie and form lines of cells. And here you can see on slide not present isogenial groups. In cartilage, fibrocartilage, predominant collagen fibers type 1. It's slide about growth of cartilages, and in general we have two subtypes of growth. It's interstitial and appositional. Interstitial growth include activates of cells of chondrocytes which produce extracellular matrix and in result increase size of cartilages. Appositional growth involves in process formation of new chondrocytes from chondroblasts. In general, chondroblasts to divide more and form in final chondrocytes, which in result include in process formation of isogenial group. I say you before about formation of isogenial group. From one chondroblast can be form one isogenial group. Okay, students, next part of lecture, bone. The first I want to say about types of bones, and in general classified according of composition and location of cells and collagen fibers. And in general, all bones to divide on two large groups. It's primary and secondary types of bones. Primary bone formed before secondary bone. And secondary bone include two subtypes. It's compact bone and spongy bone. Bones very important in process formation of a skeletal system. And similarly to cartilage, include specialized cells and specialized elements, and very important process formation of bones. All bones compact and spongy bone formed before the first formed primary and then secondary. But a primary bone in compact bone formed from cartilage, from hyaline cartilage. In bones present cells, it's osteocytes, osteoblasts, and osteoclasts. Fibers, it's collagen fiber type 1. And ground substance. To concern ground substance, it's a composition, similar to composition of ground substance in cartilages. And very interesting in bones include more neorganical salts in matrix and formed cells. And in this case, bones has very strong composition. Similar to cartilages, bone has a very important function. It's protection, it's a support, and very important function formation of hematopoietic tissue. In other words, it's bone marrow. And very important function, it's a process of regulation calcium in bloodstream. Similarly to formation of cartilage and bone, very important in this case, mesenchyme cells. Osteoblasts, similarly to chondroblasts, and lie in uh, periosteum, and its cells very important to formation of osteocytes. Osteoblasts more active to consent synthesis and produce more collagen fibers and formed more elements of ground substance, organical part in bones. In bones, near 30% is organical part and near 70% is anorganical cells. Osteocytes, similarly to chondrocytes, lie in more deeper part of bones. Osteocytes are positive of chondroblasts and chondrocytes, not form isogenial groups. Osteocytes has processes and its processes form philopodia and between cells form strong junctions. Osteocytes lie in lacuole and form composition, strong composition in osteons. It's a photo from electron microscope, micrograph. Photo show to you position of processes of osteocyte and look very attentive, very large nucleus inside in cell and active nucleus because you see more euchromatin in nucleus. Cell very active to produce some proteins but more active osteoblasts. It's very good photo from histological specimen. It's a visible position of processes and junction between processes between cells. And you see more processes in each cell present. Third type of cells in bone tissue is osteoclasts. It's very interesting cell, osteoclast, because develop and form from monocyte. And in monocyte, or normal present one nucleus, but in osteoclast may be formed from 2 to 50 nuclei in cell. It's a very active cell to concern process of digestion because more present more lysosomes which include in process destruction of collagen fibers. In present special enzyme collagenase which destroyed collagen fibers and include in process of resorption in bones. It's still very important to release minerals because process activity of osteoclast joined with active hormones. It's a special hormones which produced in thyroid and parathyroid glands. In thyroid gland present calcitonin, it's a hormone which will be decrease amount of calcium and a positive active hormone parathyroid, parathyrin, activate resorption of bones and increase amount of calcium. Its good scan show position of osteoclast near with bone matrix and osteoclast on its picture has two nuclei and good visible position of lysosomes and lysosomes produce specific enzymes which destroy of bone tissue, bone matrix and release calcium. On first slides about bone, I told about classification of bone tissue. In general, present two types of secondary bones, it's spongy and compact bones. And compact bone very strong and not present some lacunes between osteons and not present some areas, free areas. In spongy bone present more small areas without elements and in its elements lie red bone marrow. Spongy bone lie in epiphyses of long bones and include in activation process of hematopoiesis, because spongy bone is a core of red bone marrow, or in other words, it's strong stroma of red bone marrow. Compact bone, also called dense bone or cortical bone, and compact bone without some legumes, position of osteons is parallel, and we are located. Compact bone located between epiphyses lie in central part of long bones and form diaphyses. What does it mean, osteon? Osteon is a structure unit of compact bone and composed of lamels. In lamel present collagen fibers which lie parallel layer forms, and between lamels lie osteocytes. In central part of osteon present cavity, lacume, and in lacume present capilla. Cavity has name Haversel cavity or Haversel canal, and capillar a central part to provide nutrition material to bones. Its good scheme show a position of osteons and structure of osteons. In central part of osteon present blood vessel, and here are some lamels to surround blood vessel. And look very attentive. Blood vessels form some network inside in compact bone, but between osteons are not present some areas, some free spaces. Lie between osteon internal lamellar components and surround bone by external laminars, and then surround after periosteum. Very important process formation of bone, and very interesting because all processes formation of secondary bone has precursor primary bone. Primary bone is a fast bone tissue which can form from mesenchyme cells or formed on cartilage part or cartilage model. And in primary bone present location of collagen fibers not parallel and form network. In process formation of bones present intermembranous bone formation. It's a fast type formation of bones. It's a form bones provided by mesenchymal cells. Similarly to cartilageous mesenchyme cells, loose processes stay oval and form osteoblast or pre-osteoblast, one would say, which next after differentiation form osteocytes. In its case, primary ossification providing in between collagen fibers which not form parallel layers, not form lamels. Its good scheme show position of osteoblast, osteocytes, and mesenchyme cells. Here you can see mesenchyme cells with processes and next form special bone blastema. It's a group of mesenchyme cells have more short processes and form islet, and then from its cells form in future osteoblasts which produce bone matrix, and then osteoblasts penetrate in stay more deeper part of bone and form osteocytes. It's good picture because show position of osteoclast in its case. Osteoclast, you see on its picture, has 5 nuclei and lie in near with bone matrix, and in peripheral part of bone matrix present osteoblasts. In central part of bone matrix osteocytes. The next type of bone formation has name endochondral bone formation because process formation of bone include changes inside in hyaline cartilage. It's a model of long bones. In endochondral bone formation present some steps. First step is a hyaline cartilage model form, and in early period of embryonic development, a skeletal of embryo composed from hyaline cartilages, and then on model of hyaline cartilages to start form bones, and the first bone is primary. Information of bone very important capillaries because capillaries penetrate perichondrium of hyaline cartilage and mesenchyme cells formed in result osteoprogenitor cells. And osteoprogenitor cells after differentiation future form osteoblasts and then form osteocytes. Osteoblasts very active cells secrete elements of bone matrix, proteins, and include information of osteocytes. The next step for bone formation has name proliferation, and in its period, very active proliferative processes in cartilages and cells, they form columns, and it's a very important to growth of future bone. In period formation of bones, very important ossification centers formation. The first center of ossification formed in a special part of diaphysis. It's a middle part of diaphysis, and primary ossification center, its name of structure. Secondary ossification centers form more later and lie in epiphysis, and in future region between diaphysis central ossification and epiphyseal central ossification form special metaphysis. It's a part very important to growth of long bones. On its slide, very good picture show processes formation of bone, endochondral bone formation. The special name is process. The first is formation of cartilage model. It's a hyaline cartilage model. The next form ossification centers in future diaphysis, and precursor of its process is penetration of capillaries. And the next step is formation of epiphyseal ossification centers in part of epiphysis, and in a future between diaphysis and epiphysis present metaepiphyseal cartilage. On slide, metaepiphysis has specific elements. It includes three zones. The first zone has name of resting cartilage. It's a typical hyaline cartilage. It's the ending parts of epiphysis. Central zone has name proliferating zone, and it's a zone very important to growth of bones and cells. It will be flatlands, chondrocytes, form specific isogenal groups like parallel columns and form parallel columns. The short part has name, it's a part of ossification, and in its part chondrocytes will be destroyed and in future form primary specific cavity, and it's very important to formation of primary bones. On its hem, good visible position of metaepiphyseal plate. Between epiphysis and diaphysis lie a epiphyseal plate of cartilage. And it's a very interesting plate because takes information from hypophysis and very sensitivity to hormones of growth. It's a somatotropin. It's a special hormone from hypophysis which will be increase process formation of primary and in future secondary bones and provide growth of bones by long. It's photo from histological specimen. You can see position of chondrocytes and 3 zones. Secondary bone also include special areas with lamella bone, which is different. Primary bone not from lamella and secondary from lamella. What does it mean lamella? Lamella, its structure include collagen fibers type 1, which lie parallel and form strong structure. Students, its scheme show position of lamels and position of osteocytes between lamels. Black color between lamels is osteocytes, and osteocytes joined one by processes and form very strong structure. Lamel lie very strong, and between lamels not present some areas, some specimens, and in a central part of osteon present hair cell canal include capilla, and sometimes in osteon can be until 15, 16 or until 20 lamels. It's very good picture show location of lamels in osteon, amount of lamels in osteon. In central part, black color is hair cell canal, which include capilla. Students, on next slides you can see examples of tests to check the skeletal tissues. You can use its test and recognize correct answers. I hope its lecture will be important to your practical classes, to your training to practical classes. Thank you for your attention.